This is critical thinking in the Growth, Development and Genetics Lab, and here are the questions. This one is on development, and then here we have the next two questions on genetics. So let's go to this first one where you've been asked to identify the false statement. So let's look at the first one, which says the father determines the sex of the offspring. Now this is true, because if you draw a Punnett square and you look at the ovum and the sperm, the ovum has only X chromosomes, so it will only be X, whereas the sperm is either carries an X chromosome or carries a Y chromosome. So males are XY and females are XX. So when these two meet, you get XX, which means it'll be a girl. These two meet, you get XX, which will be a girl. This X meets this Y, it's a boy. This X meets this Y means it's a boy. So you have 50% chance of males, 50% chance of females. But if you look at this Y, which determines a boy, this comes from the sperm. So therefore, the father does determine the sex of the offspring. Let's look at the next statement, which says the blastocyst is the stage of development at which implantation takes place, and this is also true. So this was true too. The blastocyst is the stage of development at which implantation takes place in the uterus, and this is about 7 days after fertilization. The third one, normally the presence of HCG can be determined in the urine of a pregnant woman at the beginning of her missed period. This is true. It can be determined at the beginning of a missed period or 2 weeks, that is 14 days after fertilization. So after fertilization, 2 weeks after that, it can be determined. This is also true. And the last statement says the inner cell mass secretes HCG. Now HCG is secreted by the placenta, which is formed from the trophoblast, not the inner cell mass. The inner cell mass gives rise to the embryo proper. So since it gives rise to the embryo, it does not secrete HCG. It's the trophoblast which forms the placenta, and then the placenta secretes HCG. So therefore, this is the false statement and the answer to this question. Now let's look at the next question, which tells you that a hemophilic man marries a normal woman. And remember, hemophilia and color blindness are carried on the sex chromosomes and they are recessive. So here we have a hemophilic man. So if we look at it, the men, it will be, you represent them as X and Y, and because he's hemophilic, we can put the H over here. And he marries a normal woman, so she is normal, she is like this, and if we want we can put a capital H and a capital H to say that she carries normal genes. The lowercase h showing you that this is recessive, and this man is hemophilic. So that's how we have that. So you have to see the percentage of children who will be hemophilic and what percent will be carriers. So one important thing to remember in hemophilia and color blindness is you either suffer from the disease or you are a carrier. Very often students make this mistake thinking if you suffer from the disease, you're automatically a carrier. Now in these two conditions, these sex-linked conditions, they are different. When you suffer from a disease, you just say the person is said to be diseased. We don't call them a carrier. A carrier state here is one where a person does not suffer from the disease but carries the defective gene, and usually women are carriers, and a woman who is like this, she has one normal gene, and you can see here she has here the hemophilic gene. She's not suffering from the disease but she's a carrier. So that's what you have to determine. So let's now make a Punnett square and then we'll come to these answers here. So here is a Punnett square that we draw, and here is the woman, so she is normal. So I'm going to put this here as normal. So this is a hemophilic man. Now the man has only one X chromosome, so if he carries one defective gene, he automatically suffers from the disease. In the case of the woman, because she has two X chromosomes, if she carries one defective gene, she just is a carrier. She has to have both defective genes in order to to be suffering from the disease. So this is how it is. So let's see what we are going to get. So this X and this X will, if they meet, this is what we will get. And here again the same thing. So then here we have, and this way. So if you look at this, here we have, and maybe I should put it in red to show you this here. So here is the defective gene here, so we can see that. So here, this is a woman who has one defect of a possibility of 4 or in 100%. Here is one who has a carrier state. Here is another one who has a carrier state. So out of 4, 50%, the chances that 50% will be carriers, because these two are normal. That's what it is. 50% will be carriers. And let's see how many people will suffer from the disease. This one is a carrier, does not suffer from the disease. Carrier does not suffer from the disease. This is a normal male because he does not. He has the normal H uppercase gene. This is another normal male, a male. So in this situation, out of 4, there'll be no one who will suffer from the disease, and the chances of a carrier state is 50%. So 50% of the offsprings could be carriers, this one and this one. So then hence this is the answer to this question, and you can see all the others don't work out. So this is the way you're going to do it, whether you get color blindness or you get hemophilia. If it was color blindness, the normal gene you can write as a big C, and the small lowercase c could be for color blindness. So this is how you're going to do based on how you will form your Punnett square. Now it tells and this is how you're going to express the percentage. Let's look at the next question, which is on blood groups. So here it tells you a mother is homozygous A blood group and the father is heterozygous B. So you can write, there are different ways that you can write homozygous and heterozygous. You can write homozygous as either IA and IA, or you could, how I find it easier to write it is just as AA. This is homozygous where they are both similar. And heterozygous B, you could write it as IB and I, or how I find it easier to write it is just write BO, because two I's make the O group, and I just write it as BO. So you can do it any way you want. I'm going to use this method of AA. In other words, A group, this is homozygous A, this is heterozygous A. B, this is homozygous B, this is heterozygous B. AB will always be written as AB, and O group has to have both O's. So I just find it much easier to do it this way. So let's look at this and let's see what kind of offsprings we'll get. So we said the mother is homozygous A group, so she will have both as A. The father is heterozygous B, so he's B and O. So let's now get these. So we do this, AB, A and B will be AB, A and O is AO, and A and O is AO. So genotypically, when you say genotype, it means this genetic makeup. That means is it AA or AO or BB or BO, like that. That is genotypically. And phenotypically is what blood group are they going to be, because both AA and AO are both A. So you'll just represent it as A. So let's see this. So we can see out of this, out of 4, 50% are AB, that is genotypically, and 50% are AO. And as far as blood group goes, this group is AB is both genotypically and phenotypically are written as the same. So this is AB group and this is A group. So we can see that 50% genotypically are AB, 50% genotypically are AO, and phenotypically 50% is AB group and 50% is A group. And let's see which one of these answers shows us that. So we have 50% AB and 50% AO, which looks like this one. and phenotypically 50% is AB group and 50% is A. So this is the answer to this question, and this is how you're going to do it when you get blood groups.